All right. Hi, everyone. My name is Raghda Adwan. I'm a fourth year medical student. And today I'll be teaching you two lectures for your foundation in your physiology discipline. So I'll be teaching erythrocytes and then iron metabolism. Now, my goal is to make this as concise as possible, even though these lectures are honestly pretty complex and heavy. So I hope I can just simplify them as much as possible without taking too much of your time. Feel free to ask questions. However, I'm not sure if the chat is gonna be visible to me. So in case I don't see them, you can unmute yourself and speak if you'd like, if something's unclear. So let's just get right into it without any wasting any time. Now, to start with erythrocytes, I'll be covering the production and destruction of red blood cells, their membrane, the abnormalities that can happen, their metabolism, and how this can manifest clinically. So let's get to the basics. When you take blood, you have two components of blood. You have the formed elements, which are the non-liquid part of blood, and obviously the liquid part of blood, which is plasma, which consists of water, etc. Now, let's focus on formed elements or the cells. Now, these cells, as you guys know by now, three types: platelets, white blood cells, and red blood cells. And the entire lecture today is going to be focusing on red blood cells. So this is just to remind you of the big picture that we're just talking about the RBCs, which is just one part of blood. Now, when it comes to red blood cell structure, I'm sure you know that it's a biconcave disc, and the structure of a red blood cell is extremely important, as you'll see later on in the lecture, because any change to the structure or shape or size of an RBC can manifest in problems, and this is how we see certain diseases. So the structure and shape of it is really important. Another thing that's important is the lifespan of a red blood cell, which is 120 days. It might sound like, oh, this is a random number, but this is a very important number because red blood cells need to live through these 120 days, and any premature death of a red blood cell is a problem. Red blood cells don't have a nucleus, they don't have organelles, they don't have mitochondria. We'll talk about this again and its importance later on. Hemoglobin, of course, is the biggest component of a red blood cell, makes up 35% of its content. The cell membrane of a red blood cell, we have a whole section of the lecture on the cell membrane and its components, but of course, you'd know that surface antigens are present on the cell membrane, and these antigens are just glycolipids or glycoproteins, which act as an identification, a way to identify a red blood cell, one of which is the blood grouping. So they have A antigens, B antigens, etc. And of course, there's water, lipids, enzymes, and ions. When it comes to the function of a red blood cell, of course, it carries oxygen. It takes oxygen from the lungs through hemoglobin and carries it to the tissues where it provides tissues and cells with oxygen. And vice versa, it carries the carbon dioxide away from tissues to the lungs where it's exhaled, and it acts as an acid-base buffer to maintain blood pH in a range which is the normal blood pH. And if it deviates from this range, it becomes a problem. So red blood cells play a role in maintaining our blood pH. Now, an important concept when it comes to red blood cells and its shape is the ability for it to deform and be flexible and bend. And why is it important? You can imagine we have all these blood vessels in our body. We have very narrow capillaries and blood vessels, and so red blood cells have to be able to bend and squeeze through them. Now, the thing that allows it to be able to deform and its flexibility comes from what? It comes from the fact that it's shaped in a certain way, the biconcave disc. This shape gives it a very high surface area to volume ratio. Surface area is bigger as compared to its volume and size. So this high surface area to volume ratio allows it to be very flexible and allows it to deform. Now, the importance here is that as the red blood cell ages, why does it only last 120 days? Because as a red blood cell ages, it starts to lose its surface area. And when it starts to lose surface area, the concentration of hemoglobin inside, obviously, if you're decreasing the total volume or surface area, the concentration is going to increase because now there's less space for it to take up. Now, the concentration of hemoglobin, we have a certain value for it, which we call the mean cell hemoglobin concentration, or MCHC. So as it ages and it loses surface area, the MCHC naturally is going to increase because the concentration increases as the volume decreases. And as the concentration of hemoglobin increases, it becomes more viscous, it becomes more thick because hemoglobin is the protein that is thickening it up. So now the viscosity of the red blood cell increases. And if its viscosity increases, it becomes thicker. Obviously, naturally it would become less flexible and less able to deform itself and squeeze through capillaries. And because it's less able to squeeze and bend, it becomes damaged as it passes through these narrow capillaries and splenic pores, and this is what leads to its death. So this is the natural process of how red blood cells start to age, and they lose surface area and they become more viscous and less flexible, and therefore they will end up being damaged as they squeeze through, and this leads to their destruction in the spleen. So this is the aftermath of what happens to a red blood cell. It starts off here, it gets produced in the bone marrow, it gets released into the circulation, and then it eventually dies after 120 days in a macrophage in the spleen, for example. And this is the end product, the end pathway, which isn't an objective of this lecture exactly, but this diagram was there. Let's move on to the second part of the lecture, which is the membrane. Now, the membrane consists of proteins, lipids, and carbohydrates, and we'll be going into each one of these components one by one because they are of importance. Let's start with just an overview of the RBC membrane. So, as you guys know, as any cell membrane is, there is a phospholipid bilayer. So you've got phospholipids here, you've got phospholipids inner layer and an outer layer. Now, with the phospholipids, just like a normal cell membrane, we also have cholesterol. So the phospholipids are these with the two legs, and the cholesterol are these tiny ones with the small yellow head and shorter leg. And along with the cholesterol and phospholipids, there's also glycolipids attached on top, and of course, glycoproteins and other things. But let's go one by one. Let's start with the cholesterol. So the cholesterol exists on both faces, and the purpose of this cholesterol is to strengthen the membrane, but too much cholesterol is going to lead to not enough elasticity, so it's going to make it less deformable. So the amount of each of these components matters a lot, and your body maintains a very careful ratio of everything in a very careful amount of cholesterol, phospholipids, etc., so that it allows it to operate at maximum efficiency. So cholesterol is there to increase membrane strength. That's the point. Now, when it comes to phospholipids, there are different types of phospholipids, and some of them mostly exist on the outer layer, and some of them mostly exist on the inner layer. So I know these names are a bit of a mouthful, but they are pretty important, especially one of them, which I'll get to in a minute. Now let's look at the outer layer. We have phosphatidylcholine and sphingomyelin. These exist on the outer membrane. Okay, the inner membrane, you've got phosphatidylethanolamine and phosphatidylserine. Now I want you to focus on phosphatidylserine, which is the most important one here. So, as we said, phosphatidylserine exists where? On the inner membrane. What happens as the cell ages? As the cell ages, or it becomes damaged, or there's inflammation, there's an infection, there's something going wrong, or just normal aging. Either way, phosphatidylserine has this tendency to flip onto the outer membrane. So it goes from the inner membrane to the outer membrane. Now, why does this matter? So what if it flips on the outside? This matters because it's not supposed to be on the outside. It's supposed to be on the inside. So it acts as a signal when it becomes on the outside. Now, other cells can see it, obviously, since it's on the outer membrane. Other cells can see it, and some cells can recognize it because it's a signal. And specifically, macrophages in the spleen, they have a specific receptor for phosphatidylserine. So when phosphatidylserine goes out to the outer membrane, and it passes through the spleen, in the blood vessels in the spleen, the macrophages in those blood vessels in the spleen, they have a receptor, and they're going to be able to recognize it, and then they bind to it because now it's on the outside, and they'll remove it from the circulation. And this is part of how RBCs end up dying. So this can happen as the normal aging process of the red blood cell, or it can happen when there is damage or inflammation or infection. CRP is C-reactive protein, which is a marker of inflammation. So when anything is going wrong, it can result in red blood cells dying prematurely, or in the normal aging process. Now, we've covered cholesterol, we've covered phospholipids. Now, the ratio between them is really important, and the body maintains a very careful ratio, as I said, because they can make the membrane stronger, they can make it less elastic, they can, that ratio is important. Any changes in this ratio are linked to some abnormalities, such as an acanthocyte, which is this type of cell, which has like spikes coming out of it, or a target cell, where you've got a central area of all of the hemoglobin going in the center while there's a surrounding area of pallor. Now, you don't need to memorize these specifically. It's just an example to show you that generally any alteration in the normal membrane will result in abnormalities that you're going to be able to see on a red blood cell, such as these two. All right, now the next component are glycolipids. Glyco meaning sugar, lipid meaning lipid. So you've got sugars attached to the lipids, and these act as antigens. So, as we said earlier, the ABO blood group antigens, they are just a marker. So they show you that, okay, this is blood group A, this is blood group B. And this is how our red blood cells are identified in our body through these glycolipids or glycoproteins. Now, the glycocalyx is the glycoprotein polysaccharide sugar layer. So the whole layer that covers the red blood cell, because of these glycolipids and glycoproteins, is called the glycocalyx. Why is it important for us to talk about it? Because the glycocalyx is naturally negatively charged. So it makes the entire red blood cell negatively charged. This is important. Why? Because it prevents attack from microbes such as bacteria and viruses and parasites, number one. Number two is that it prevents any mechanical damage by happening because naturally, if red blood cells are negatively charged, they're going to slightly repel each other and they're not going to start to adhere or stick to each other, which we don't want. And so, because they're negatively charged, they repel each other, and this prevents mechanical damage between red blood cells themselves or the red blood cell in the endothelium of the blood vessel. So, it's just the layer of the glycocalyx layer is important for these purposes. Okay, we're done with the lipids. Please let me know if I'm going too fast. I just don't want to, talk too much about any specific part of this lecture, so I don't take much of your time because you guys have enough on your plate for foundation. So please let me know if I'm going too fast for your liking. If you want me to slow down, if you want me to cover anything once again. But if everything's okay, I'll continue. Now, the proteins. We covered the lipids. Now let's go on to the proteins. So as you can see, again, this is a different diagram, but we've got phospholipids in the bilayer. With the phospholipids, of course, we have proteins. Now, don't get overwhelmed with these weird names. We'll go through them one by one, but they are important when it comes to, even clinically, it's important. Now, you can see here we have these two long pink tubes. These are alpha and beta spectrin. We have actin right here, the cylinder. We have protein 4.1. We have this green one, ankyrin. And we've got band 3 protein, as well as these glycophorins, which are the integral membrane proteins. So this is the overview. I'll talk about each one and a brief function of what it does. But you can already tell that some of these are aligned vertically and some of them are aligned horizontally, which we'll come back to in a minute. So let's start with spectrin. These long pink tubes, spectrin is a cytoskeletal protein. You can see how it's aligned. It's not really part of the membrane. It's more part of the cytoplasm, but then it's anchored to the membrane. This is the cytoskeleton. Cytoskeleton is the part in the proteins in the cytoplasm which give the cells structure. So spectrin, alpha and beta spectrin, they are cytoskeletal proteins that are aligned to the membrane in an antiparallel manner. Next up is ankyrin. So you can see here, ankyrin is obviously binding to spectrin. You can see they're bound there. And then ankyrin is also bound to band 3 protein. So band 3 protein is integral protein and it secures spectrin. So you see how it's like band 3 protein is holding hands with ankyrin, which is holding hands with spectrin. So they're all anchored together. They're all held together between the cytoskeleton and the membrane itself. This is what gives things structure, and as well as acting like an anion exchanger, but I don't think this is too important. Next up is actin. Actin is also obviously bound to spectrin, and it's also bound to protein 4.1, which is attached to it, as well as then being bound to the glycophorin in the integral, which is an integral membrane protein. So you see how, again, glycophorin is held by actin, which is held by protein 4.1, which is held to spectrin. So everything gives each other structure. So we covered band 3 protein, ankyrin, spectrin, actin, protein 4.1, and the glycophorins. Now, if you just look at this diagram, you can see how things are aligned. There's a vertical interaction, which is just vertical stability, let's say, instead of interaction. The vertical interactions are creating vertical integrity or stability. And this is ensured by, which is a key player here, which is ankyrin, and of course, the transmembrane proteins. The horizontal integrity is ensured by spectrin. As you can see, it's in the cytoskeleton and it's horizontally laid. So it's ensured by spectrin and other cytoskeletal proteins. These are the proteins in this red blood cell membrane. Now, are there any questions so far? Or is everything okay? Okay, I'm going to assume that everything is all right. Can you explain the two phospholipids again? Yes, I can. Let's just quickly go back. 1 second. Okay. This part, right? Whoever was asking. Go ahead. Yes, okay. I'll explain this again. So, phospholipids are in our red blood cell membrane, just as they are in the membrane of all cells. We divide them in the red blood cells based on the phospholipids that are in the outer membrane versus the phospholipids that are in the inner membrane, because they're supposed to be where they're meant to be. This is their purpose, is to be in the inner membrane or in the outer membrane. We have two in each one. Two phospholipids in the outer layer, phosphatidylcholine and sphingomyelin, and two phospholipids in the inner layer, phosphatidylserine and phosphatidylethanolamine. The most important phospholipid here is phosphatidylserine, PS, phosphatidylserine, okay? We said it's supposed to be in the inner membrane. When things go wrong, such as damage or inflammation or infections or just normal aging, phosphatidylserine is going to flip to the outer membrane. It's going to move. It's going to go from being an inner membrane phospholipid to an outer membrane phospholipid. Once it's outside, now it's facing all the other cells in our body. The cells that can detect it are the macrophages in the spleen. The macrophages in the spleen have a receptor. They're going to look at phosphatidylserine on a red blood cell. They're going to go like, oh, this is not supposed to be on the outside. I'm going to go and eat this red blood cell and take it out of the circulation, basically destroying the red blood cell. So this could be a normal part of the aging red blood cell. So it has reached 120 days. Khalas, it's time to die. And it dies by phosphatidylserine going to the outside, or because of damage to the red blood cell or inflammation, it's a response. So it's going to flip outside as a response to any damage to the red blood cell and therefore die prematurely. No, it's not 200 slides. It's just I don't have animation, so I divide the slides up. It's about 40 slides per lecture. I'll explain band 3 in a moment. Where are the slides not in the drive? I'm not sure. I think, please, the hosts, can you ask the heads if they can share it with the first years? Spectrin is for horizontal movement. Yes, it is. Spectrin is a horizontal protein. So let's go back to the proteins once again. Okay, spectrin is is a part of the horizontal interactions because it's laid down in the cytoskeleton. It maintains horizontal structure or integrity. Band 3, to the person that was asking, it's an integral protein. Integral protein means it's integrated to the membrane, and therefore it's able to hold other things onto the membrane. And so this is what makes it integral. And it secures spectrin to the membrane because it's also anchored to ankyrin. And it also acts as an exchanger for ions, but I don't think that's a vital piece of information for you. I hope everything is clear now. What's the function for protein 4.1 and 4.2? These proteins, they are just there as extra support. You see how it's just anchored. It's just another addition to all the proteins being anchored together. The whole point of these proteins, you don't need to know, oh, protein 4.1 is anchored to this, which is anchored to. You just need to understand the idea that every protein, the proteins are attached to each other in order to maintain structure. You can't have things just existing in the red blood cell doesn't have structure without these proteins that are holding everything together. I assure you it's not 200 slides. It's just you can see how I divide the slides one by one, which makes it look like 200, but it's not. Okay, are there any other questions? I'm happy to answer them. If not, we can move on. All right, so I'm going to take that as a no. All right, let's move on to abnormalities. So just a reminder of the normal morphology of a red blood cell. It's a biconcave disc. What is it maintained by? Spectrin and ankyrin, as we already just discussed, and the other membrane proteins, but these are the most important. The advantages are the surface area, the flexibility, and the fact that it reduces membrane tension. So before we get into the different morphologies or the different appearances of red blood cells when they're abnormal, just remember how we're looking at them. We're taking a blood smear, looking at them in a microscope, and then we're looking at the smear of blood. Now, just one thing before we go into each of these abnormalities, you don't need to know the cause. the diseases which give you each of these abnormalities. But I want to go through them so you understand that everything that we talk about, it makes sense. It makes sense when you consider the name of the abnormality and why it's happening. And then you later on, when you study these diseases, you can link it to why it actually happened. But now, because this was part of your lecture, and I don't want to miss anything that was discussed in your lecture, we're just going to go through them quickly at face value and what they mean, just to have an understanding of why they're named the way they are. So any variation in size of a red blood cell is called anisocytosis. Any variation in shape is called poikilocytosis. So a red blood cell could be abnormal in size or it can be abnormal in shape. First one, that is a normal red blood cell, okay? That's exactly what it's supposed to look like. This one, as you can tell, it's bigger, much bigger than it's supposed to be, and this is abnormal. This is a macrocyte. Macro meaning too big. This one, what does it look like? It kind of looks like a target cell. I mean, it kind of looks like a target because you have the central area where there's concentration of hemoglobin, and then around that, it's pale. And then it goes back to its normal color after that. So this kind of looks like a target, which is why we call it a target cell. This one right here, it has a slit like pale area in the center. This is called a stomatocyte. Stoma means a slit like opening. This one looks like a pencil, so we call it a pencil cell. This one right here, you see how there's like spikes coming out of it? This one, we call it an echinocyte because this is echinos in Greek is the name for a sea urchin. So you can see that they just named it. It's kind of it's very straightforward the way they name things once you understand where it comes from. So this is an echinocyte. You can see it in certain diseases. The next one is an acanthocyte, which is, again, it's sort of like membrane blobbing off. So it creates this cell called an acanthocyte. This one right here, you see how it's smaller and also darker? This one we call a microspherocyte. So it's smaller than it's supposed to be. And it's very spherical in shape, which means all the hemoglobin is packed in, making it spherical, making it darker in color. So microspherocyte. This is a burst red blood cell, which has burst into fragments. So these are fragments. This is sort of a more elliptical shape of red blood cells, so it's called an elliptocyte. And this one right here is in the shape of a teardrop cell. This one is in the shape of a basket cell. But honestly, you're not really going to use this name or identify this much because even till fourth year, we've never spoken about a basket cell. This one, which is important, you guys are going to talk about this a lot. What does it look like? A sickle. So we call it a sickle cell, and this is seen as sickle cell anemia. This is very high yield for you later on. And now lastly, you see how this is very small and paler than normal. This is a microcyte, which is also important when, I'm sure you're going to take this in your hematology lectures. Now, I'm not saying this for you to memorize every single shape and every single cause. I'm showing you because it was in your lecture, number one. Number two, it's pretty straightforward when you understand why something is named the way it's named. Okay. Do we have to memorize the causes? No, you do not. No, you do not have to memorize the causes, but this table was in your lecture, so I had to cover it quickly, but you do not have to memorize the causes. Isn't it hypochromic? Yes, it is hypochromic. Very good. Because the color is paler than it's supposed to be. So it's a microcyte, that's the red cell abnormality, and it's also hypochromic, which iron deficiency, as I'm sure you know by now. So microcyte is the name of the cell, also hypochromic. All right, now let's put this into perspective. Why are we studying about the proteins and the membrane and the ankyrin and spectrin? Because there are some disorders, genetic disorders, where there are abnormalities in these proteins. And any abnormality in these proteins is going to cause problems. One of these disorders is hereditary spherocytosis. Hereditary meaning it's genetic, not acquired. And spherocytosis, obviously, you've got spherocytes, lots of spherocytes, meaning spherocytosis. Sphere means spherical. Now, red blood cells are already kind of circular in shape, but they're not a perfect sphere. They're not supposed to be. This is, I believe, this is what it's supposed to look like, sort of. But you see how these are very dark. First of all, there's no area of normal pallor in the middle. And they're dark and they're small, slightly smaller and very spherical, very round in shape. This is not how your blood cells are supposed to look. So these are spherocytes and they're abnormal. So how is this happening exactly? Firstly, here's hereditary spherocytosis is hereditary hemolytic anemia. Hemolytic meaning hemolysis, which is heme blood, lysis bursting or breakdown. So it's a breakdown or a lysis of blood cells. Obviously, when you have lysis or breakdown of blood cells, you're going to get anemia because you're going to decrease hemoglobin naturally. So how does this happen, and why do they become spherical in shape? They happen because of a deficiency or abnormality in the proteins we discussed. So ankyrin, most commonly ankyrin, and also spectrin, band 3, or protein 4.2. So bottom line is there's a problem or abnormality in the proteins that are in the red blood cell membrane. Now, when you have a defect in these proteins, you're going to have a defective interaction between the cytoskeleton and the lipid bilayer. So you're going to go from having a normal biconcave red blood cell to defective membrane, which is going to cause it to lose membrane because it's defective. So it's going to start to slowly lose the membrane itself. Now, loss of membrane is going to cause a lower surface area to volume ratio. It's going to lose the biconcave shape, and it's going to start to get smaller and gets more spherical. Now, as these spherocytes circulate in the spleen, in the vessels in the spleen, and in the reticular endothelial system, these spherocytes will get trapped in the microcirculation because they have an abnormal shape, number one. And number two, they don't have flexibility or deformability. And as a result of those things, they're going to die prematurely because of splenic macrophages or just hemolysis. And this is going to result in a hemolytic anemia. So this is one abnormality that can happen to your red blood cells based on the proteins in the membrane. And this is a genetic disorder. And now this is actually pretty important. We even third year we took this again, so I'm sure it's high yield to you guys as well. Now, that is one genetic disorder. Other abnormalities, as I'm sure you know, anemia, low hemoglobin, low red blood cells. The types include iron deficiency or vitamin B12 deficiency, which is going to manifest in different ways. We have polycythemia, which is a very high red blood cell count and higher than normal. This could be due to hypoxia because your body sees that it doesn't have enough oxygen, so it tries to make more blood cells, or because it could be because of a genetic cause. Or shape disorders, as we just spoke of, spherocytosis. It's not supposed to look like a sphere, and we also have sickle cell anemia, and all the other abnormalities that I showed you. So the bottom line here is to understand we've got red blood cells. The membrane has components which are very important. The amount of those components is important as well as their effective shape and existence. So any abnormality in the membrane is going to manifest in, first of all, you're going to be able to see them in the ways that I showed you, and also it's going to affect the lifespan of the blood cell. It's going to start to die prematurely or more than it should, and this will affect the body in different ways. Let me see someone said in the chat. What's cythemia? Cythemia. So poly is too much. Cythemia. Erythrocytes. So cyte is blood cell. Emia is the blood. So when you've got too much blood cells in your blood, which is polycythemia. Any more questions? I hope everything's clear. If anybody has any issue with what was covered, I don't have a problem with covering it again. If not, we'll move on. Okay, great. All right, so next part of the lecture is red blood cell metabolism. Metabolism is just how your body makes ATP and uses it. It's just the normal function of your body. Without metabolism is the way your body functions in general. It's all the chemical reactions that are happening to allow your body to function. Now, red blood cells have specific ways of conducting metabolism because they're not like other cells. How are they not like other cells? If you guys remember, they don't have organelles. They don't have a nucleus. If you don't have organelles, you don't have mitochondria. If you don't have mitochondria, you cannot carry out aerobic respiration, which is the chemical reactions that are happening inside the mitochondria to make ATP. So how come the red blood cells can maintain function without ATP? They do have ATP. They do make ATP. They do it in a different way than normal cells because they lack mitochondria. So they're going to rely on a different process called anaerobic glycolysis. You guys are going to take this entire pathway in detail when it comes to mole. So I'm just going to give you an overview for what you need to know in terms of red blood cells. ATP, as you should know by now, is the main energy currency for any cells or in our whole body, especially red blood cells. The role of ATP is to maintain volume, maintain shape and flexibility, as well as protect the red blood cell from oxidative damage. So it slows down oxidation of hemoglobin and membrane proteins from any type of free radicals or such as peroxide and superoxide. So this is going to preserve hemoglobin function and membrane integrity. Okay, so ATP is vital for these purposes. If you don't have enough ATP, this will also manifest in its own problems and pathologies. ATP is also important for transportation of ions. So we've got transport pumps. Sodium potassium ATPase for one. It balances sodium potassium concentration and maintains osmotic stability. So when you maintain sodium and potassium concentrations in and outside of the cell, you maintain water where water goes. So if you got too much sodium or potassium inside, water is going to run in, the cell is going to burst because too much water, and vice versa. So the balance of these ions is really important. What maintains the balance? ATP. The second pump is calcium ATP, so it keeps calcium stores low, so you don't have too much calcium inside, and also prevents osmotic damage as well. Another thing is aquaporin. Aquaporin is a water channel which allows movement in and out with any osmotic changes. So that's how water can go in and out through aquaporin. And it works with potassium, sodium, and calcium to maintain osmotic balance. Another important note about hereditary spherocytosis, you also might have an abnormality in aquaporin 1, which adds to the problems they already have in hereditary spherocytosis. So, with that being said, we said ATP does all of these things. So what happens if ATP is lost? If ATP is lost, you're going to have failure of sodium, potassium, and calcium pumps. And if you have failure of those, sodium and calcium are going to build up inside the cell. And where sodium is, water will follow. So water follows into the cell. If you've got too much water overloading the inside of the cell can't take it anymore. It's going to swell and rupture and burst. You're going to have hemolysis. This is called osmotic hemolysis. Hemolysis being the fact that it's rupturing, osmotic meaning because of water. And this is because of ATP imbalance. So just keep this in mind when we talk about how ATP is made. If the process of making ATP in a red blood cell is abnormal, you're going to have water coming in, too much water coming in, osmotic hemolysis, okay? Now, another important note when it comes to ATP and metabolism is sickle cell. So when you reach a stage of deoxygenation, or when the blood cell loses the oxygen, the hemoglobin S, which is a type of hemoglobin found in sickle cell disease, I'm sure you guys took this in a different lecture, but hemoglobin S is going to start to polymerize. It's going to clump and stick together. And this clumping and sticking together is going to form the sickle shape of the red blood cell in sickle cell disease. This sickle shape is going to increase membrane permeability to calcium. If there's too much calcium, as I just mentioned, it's going to cause hemolysis. So the whole point of this is to show you that a red blood cell needs to be able to make ATP properly. If you cannot make ATP, you get all of these problems. So now, how does it make ATP? As we said, it lacks mitochondria. So what does it do? It relies on the cytoplasm. And the function, there are a few pathways, there are three pathways which we're going to discuss. Each one is vital in making ATP, as we just mentioned for the purposes that we in the previous slide. Also for making NADPH, which is another molecule used for the survival of the RBC, and also to maintain hemoglobin's oxygen affinity, the binding of oxygen to hemoglobin and the carrying of oxygen and releasing it to tissues through this molecule called 2,3-bisphosphoglycerate. I know this is a bunch of new, like what's NADPH and what's 2,3-BPG and what's all this. All of this is going to be explained in detail in mole. You're going to take all the pathways in mole. Here, it's mentioned in your lecture, and it's important to note because red blood cells, they rely on these pathways in order to maintain function. So I'm giving you an overview of the purpose and how it does it and what can go wrong, but without the tiny, tiny details. All right, before we get into LC. Someone has sickle cell, do all their blood cells look abnormal? Is it just a percentage? It depends on how severe their disease is. So you know how it's genetic, there's two alleles. They could have HBS, two forms of HBS, which means this is the most severe form, and most of their blood cells are going to be sickle shaped. Or they could have HBS with HbA, so they could be heterozygous, and this could have a less severe disease. So it depends on their genetics. It depends on the severity of the disease. So it depends. What do you mean by mature RBC lack mitochondria? So immature RBCs have. The red blood cells, once they have left the bone marrow, they don't have mitochondria. That's what you need to know. Blood cells, like right now, if I look at your blood and I take it out, they don't have mitochondria. In the development phase, at some point they might have certain organelles for their development, but the point is they don't have like normal red blood cells don't have mitochondria, mature ones. When they just die, if most of their RBCs are sickle shaped, of course this is going to it can cause sickle crisis. It can cause severe pain. It can come in various clinical manifestations. But the fact that they're sickle shaped isn't an immediate death. It's not fatal, immediately fatal, because doesn't mean that the person's going to die because their RBCs are sickle shaped. It can be managed and it can be, you know, dealt with. We can discuss. You're going to have a lecture on sickle cell disease and its severities and its genetics and all of the components that play into it. It is very interesting, and I understand where your questions are coming from. But they're not. It's not a death sentence, is what I was looking for. Okay. Okay, tamam. I'm going to continue if there are no more questions. No problem. I hope this made everything clear. All right. Now, let's get into these metabolic pathways that I just mentioned. There's three of them. Number one, the glycolysis or Embden-Meyerhof pathway. Number two, the pentose phosphate pathway or HMP shunt. And number three, Rappaport-Lewis-Bring shunt. Now, I know you're going to look at this and go like, what in the world? Because honestly, this is biochemistry. Now we're talking about biochemistry. I'm going to try to simplify things and keep it focused on why this matters in red blood cells. So don't overthink it and don't get lost in the details here. You're going to take this in detail later on. But I have to cover them because they are in your lecture. So just try to stay with me here. Now, glycolysis. I'm not sure if you guys took this yet, but you're going to take it in Mo. Glycolysis is the process by which we take glucose from our food, we break it down, and we make ATP. Part of glycolysis happens in the cytoplasm. So it becomes the main source of ATP in blood cells because they don't have mitochondria, they only have cytoplasm. So they're going to rely on glycolysis, specifically the anaerobic form. Per every glucose molecule, we end up with ATP, the energy we need. Byproducts are NADH and lactate. Now, why is this important? Because glycolysis is a bunch of chemical reactions. What is running these chemical reactions? Enzymes. And what can happen to these enzymes, genetically speaking? You can have enzyme deficiencies. You can just lack some enzymes. They're not there. And so if there is a defect in an enzyme of glycolysis, what's going to happen? You can't produce ATP. What's going to happen if red blood cells can't produce ATP? They're going to end up with hemolysis. This is what I want you to understand. All of these are chemical reactions. Chemical reactions are run by enzymes. If the enzymes are defective or deficient, you're not going to be able to produce ATP in this pathway. If you don't produce ATP, you are going to have a problem, hemolysis. Which enzymes most commonly in glycolysis? There's two of them. Most of the deficiencies are the pyruvate kinase deficiency. The other one is phosphoglucose isomerase. I think this is the one that you should memorize if you were to memorize one of these names. Let me show you just quickly. This is all of the metabolic pathways together. This is glycolysis, extending down here. This whole line, follow my cursor. Pyruvate kinase is this enzyme. So a defect in pyruvate kinase is going to mess up this pathway. But again, please don't be overwhelmed by the picture I showed you. Just focus on what I'm explaining. That is glycolysis, okay? The second pathway is pentose phosphate pathway, PPP, or HMP shunt. It's the same thing, they just have different names for some reason. This pathway, we said the previous one was to make ATP. This one is to focus on making NADPH. NADPH is a chemical. It's a molecule which we need. Why do we need it? Now, NADPH here, it is part of this other reaction pathway. NADPH is needed because it helps in reducing oxidized glutathione. Oxidized glutathione is GSSG. So NADPH is needed. See how the arrows are going together? NADPH is needed in order for GSSG to become GSH, which is oxidized to reduced, okay? NADPH is needed to reduce the GSSG into reduced glutathione. Why do we need reduced glutathione? Because reduced glutathione is able to take on any oxidative damage that happens instead of it damaging red blood cells or hemoglobin. So any oxidant damage, superoxide, peroxide, free radical species, these are the things that happen when there's inflammation or infection or disease in your body. This oxidative damage, because of whatever reason, is going to come, and the reduced glutathione is going to be oxidized in order to prevent the damage to anything else. It's going to take on the damage, okay? Take on the oxidation. This is normal. This is how your body responds to oxidative damage normally. And if there's a defect in this pathway, if there's no reduced glutathione, the oxidative damage is going to affect what? The red blood cell or the hemoglobin. Obviously, you don't want that. You don't want the red blood cell to be damaged because of oxidation. You don't want the hemoglobin to be oxidized and form abnormal hemoglobin. This is going to severely affect your body. You need the hemoglobin and red blood cells. So because of this, reduced glutathione is going to take on oxidative damage and become oxidized. This is normal. What do we need for this recycling of reduced glutathione? We need NADPH. We need NADPH to maintain reduced glutathione. Okay, now where do we get in NADPH from? That's where pentose phosphate pathway comes in. Pentose phosphate pathway is regenerating all the NADPH. It regenerates NADPH in order to regenerate reduced glutathione to detoxify oxidative damage. Okay? Before I get into the enzyme deficiency, let's make sure this was clear. Can you repeat, please? Yes, I will repeat. How does NADPH prevent oxidative damage? Okay. So let's go back to this right here. Look at this right here. You have GSH is reduced glutathione. This is reduced glutathione. GSH is going to be oxidized by oxidative damage. Oxidative damage is caused by inflammation, infection, disease. You have oxidative damage all the time. Even without the disease, you have oxidative damage in your body in some aspect. Oxidative damage is going to be responded to by the reduced glutathione. Reduced glutathione is going to become oxidized instead and take on the damage instead, and it becomes oxidized into oxidized glutathione. Then oxidized glutathione, we need to regenerate more reduced glutathione so that we can keep responding to the oxidative damage. To regenerate the reduced glutathione, we need NADPH. We need NADPH. It reduces it and regenerates it. I know how this can get confusing, but just bear with me, okay? NADPH is needed in order to regenerate this one. NADPH is needed to regenerate reduced glutathione. If we have reduced glutathione, we can respond to oxidative damage without harming our body, okay? Without NADPH, you're not going to be able to get reduced glutathione. Without reduced glutathione, you're not going to be able to manage oxidative stress. And so this oxidative stress is instead going to go and damage hemoglobin and red blood cells, which we don't want. This is why NADPH is important in this process. Where does it come from? It comes from the pentose phosphate pathway. I hope that cleared it up. Thank you. Okay. You're welcome. Okay. Now, how can this become a problem? When we have a deficiency of the enzyme, which gives us NADPH. What is this enzyme? G6PD. This one you're definitely going to take somewhere else, I think in maybe foundation or somewhere else in first year. G6PD is a very important enzyme because a deficiency of this enzyme means you are unable to regenerate NADPH. Okay, so let's start again. You need NADPH to have reduced glutathione, to recycle reduced glutathione. You need reduced glutathione or GSH in order to handle your oxidative stress in the body. Where does NADPH come from? It comes from the pentose phosphate pathway. Which enzyme makes NADPH? G6PD, glycerol 6 phosphodehydrogenase. So this G6PD is regenerating NADPH. NADPH is regenerating glutathione, reduced glutathione. Reduced glutathione is taking on the oxidative stress in your body in order to maintain everything being normal and the oxidation not going to your hemoglobin or red blood cells. A deficiency in G6PD is going to lead to impaired production of NADPH. So your red blood cells, they don't have a defense mechanism, which is the glutathione. They're going to be susceptible to oxidative stress. What is oxidative stress? As I said, infections, inflammation. There are certain drugs that are that give oxidative stress, like antimalarials. And if you don't have G6PD to regenerate NADPH, any oxidant stress like these things can go and oxidize hemoglobin, which will denature hemoglobin, and you're going to end up with abnormal hemoglobin. It's going to damage the RBC membrane, and it's going to lead to hemolytic anemia. Please feel free to ask. Whoa, it just clicked for me. Thank you. You're welcome. I'm really glad that it clicked because I know this can get confusing. So I can repeat it again if anybody needs clarification. Again, please. Okay. Okay. Don't worry. Okay. Let's restart. Let's start over. Okay. So look at this pathway right here. This is how your body recycles glutathione, reduced glutathione. GSH is glutathione, okay? Reduced glutathione. Your body needs to recycle glutathione. Why? Because glutathione is going to take on oxidative damage or oxidative stress in your body, oxidation, through superoxides and peroxide. These superoxides and peroxide and oxidative stress needs to be neutralized somewhere. Your body needs to neutralize it. How does it neutralize it? By this molecule called reduced glutathione. When the oxidative damage comes, it will oxidize the glutathione, the reduced glutathione, into oxidized glutathione, GSSG. This process is normal. This is how your body is defending itself from oxidative stress. Now, this isn't like an endless supply of reduced glutathione. We need to regenerate it again and again. We don't have an endless supply. You need to get back to your reduced glutathione. You need to reduce it again. How do you reduce it back to reduced glutathione? You need NADPH. You see the arrows here. It's going from GSSG to GSH. At the same time, NADPH is being used. NADPH is needed for this reaction. NADPH, where does it come from? You need to also recycle it. You don't have an endless supply. NADPH is coming from an enzyme called G6PD, glycerol 6-phosphate dehydrogenase, I believe. So this enzyme is regenerating your NADPH. Your NADPH is needed to regenerate reduced glutathione, which is GSH. So NADPH is needed for this reaction. So what does reduced glutathione do? What's the purpose of everything we just said? Reduced glutathione is there to take on oxidative stress so this doesn't damage your body and your blood cells and your hemoglobin. This is bottom line here. We need this molecule in order to be oxidized itself instead of the oxidative stress going, oxidative damage going and damaging your hemoglobin or red blood cells. See how it's 3 arrows? This is the first way. If there's an abnormality in this pathway because of a deficiency in G6PD, the oxidative stress is going to go and oxidize hemoglobin and red blood cells, which you don't want. This is not normal. Okay? This is NADPH. Thanks. Are you 100% sure you got it? What about Muhammad? Please tell me if you got it. All right, perfect. Alhamdulillah. Again, this is just us talking about this pathway, which is called the pentose phosphate pathway or the HMP shunt. This is where we get our NADPH from, and it's where G6PD functions. Alhamdulillah, now that everyone, it's clear for everyone, I'm going to move on to the third pathway. We're almost done with the first lecture, I promise you. The third pathway is the Rappaport-Lewis-Bring shunt. Now, this is the only time you're ever going to hear about this. It's not going to come up in MOL. It's not going to come up in second year or third year or anywhere. It's a very specific pathway for blood cells. It was covered in your lecture, so I'm going to have to explain it. Very simple. It's just a form of glycolysis that is unique to red blood cells, and it's there to produce 2,3-BPG, 2,3-bisphosphoglycerate. Why is 2,3-BPG needed? Because it binds to hemoglobin. It's going to decrease oxygen affinity. It's going to go to hemoglobin, and the hemoglobin is going to go like, khalas, I don't want the oxygen anymore. Oxygen is going to detach. And we want this because oxygen needs to be released to the tissues. We don't want hemoglobin to hold on to oxygen forever. Hemoglobin needs to let go of the oxygen. How does it let go of oxygen? Because of 2,3-BPG. Simple? It's pretty straightforward. This one doesn't need a lot of thinking. It's just Rappaport-Lewis-Bring shunt is used to make 2,3-BPG, which allows oxygen to detach from hemoglobin. This is an overview of all the pathways, the 3 that I just talked about. EMP or glycolysis, this one. Pentose phosphate pathway is this one off to the side, which is generation of NADPH. The third one is Rappaport-Lewis-Bring shunt, which is used to make 2,3-bisphosphoglycerate, 2,3-BPG or DPG, that is used to regulate hemoglobin affinity for oxygen. Is this clear? Can you repeat glycolysis? Yes. Oh, sorry. So glycolysis is a pathway by which the red blood cell makes ATP. ATP is the main purpose of glycolysis. If there's a defect in the enzymes of glycolysis, you're not going to be able to make ATP. If you don't have ATP, you're not going to have the ion pumps that we mentioned earlier, they're going to become defective. You're going to have imbalance of ions, sodium and calcium and potassium. If there's too much sodium and calcium inside because of lack of ATP, water is going to go inside the cell. Too much water inside the cell is going to make the cell swell and rupture, leading to hemolytic anemia or hemolysis. This is why enzymes in glycolysis are very important. What are the enzyme deficiencies that can happen? Most commonly, pyruvate kinase. But you guys will take all of glycolysis. It's a 10-step pathway. You'll take it in mole. Right now, I just need you to understand how this is important for red blood cells. Does NADH or NADPH come first? NADH and NADPH are two different molecules. They have different functions. They're different because of the presence of the P. So it's not that one of them comes first or the other comes second. NADH is different than NADPH. See how this is NADP, not NADH. Don't worry about NADH. Just worry about NADPH for this lecture. One of them is just a reduced form, one of them is oxidized. So it's not like one of them comes first or second. They're just different forms of the same molecule. This is the picture that was used in your lecture. Do not worry about memorizing anything in this picture. This is just overview. This is glycolysis, EMP. This is the pentose phosphate pathway, or HMP shunt, the second pathway, which is producing NADPH. And this right here is rapid lobe brain. Honestly, I tell you, just ignore this picture. It doesn't matter. Just focus on what I explained earlier. But I included it because this is the picture in your lecture. And these are the enzyme deficiencies that can happen that the doctor highlighted, but I already mentioned them. So G6PD, glucose 6-phosphate dehydrogenase, pyruvate kinase, and glycolysis. And this one, I don't think you need to worry about, to be honest. Glucose 6-phosphate isomerase. Is everything clear so far? Before I move on to the last part of the lecture. Okay, great. I hope everyone is following well. Okay, we're almost done with the erythrocytes lecture. Just bear with me for the last part. So we have another important enzyme. This is not related to metabolism or glycolysis or these things. That's why I separated them. The last one is called methemoglobin reductase. This pathway is there to solve a problem. What's the problem? Oxidation of hemoglobin. Remember how I said earlier, we need NADPH to get glutathione so that we can combat the oxidative damage. What if hemoglobin gets oxidized? This is a problem and we need to solve it. We have a different way to solve it. If hemoglobin gets oxidized, which is what we don't want, but if it does get oxidized, it goes from being Fe2 plus to Fe3 plus. It goes from Fe2 plus to Fe3 plus. Fe3 plus is useless hemoglobin, okay? We don't want Fe3 plus hemoglobin because it's not able to carry oxygen the same way that Fe2 plus is able to. So this Fe3 plus hemoglobin is called methemoglobin. Methemoglobin. This is functionally useless, and we need to reduce it back to normal hemoglobin. From Fe3, we need to reduce it from Fe3 plus back to Fe2 plus to reverse the oxidation. How do we do this? We have a pathway with an enzyme. It's called methemoglobin reductase. Reductase means it reduces it from Fe3 plus to 2 plus. Methemoglobin is being reduced. This enzyme plus NADPH is going to reduce Fe3 plus back to Fe2 plus, and this keeps everything normal and safe. It's simple. This is the diagram in your lecture. You've got hemoglobin Fe2 plus, which is good, normal. If it gets oxidized, it becomes methemoglobin, which is Fe3 plus. Iron 3 plus, iron in the 3 plus state. This methemoglobin, we have ways to take it back to Fe2 plus, which is by this enzyme, methemoglobin reductase, which will convert it back to Fe2 plus hemoglobin, which is normal hemoglobin. Now, why is this relevant and why are we talking about this? Because some people, they have a genetic deficiency of methemoglobin reductase. They cannot reduce Fe3 plus back to Fe2 plus. This is called hereditary methemoglobinemia. Methemoglobinemia means methemoglobin in the blood. And it's hereditary. It could be genetic, but it can also be acquired. So hereditary form is one form, acquired form because of drugs or chemicals or certain things that can cause methemoglobinemia. The reason why I brought this up is it's in the lecture, number 1. Number 2, when you have methemoglobinemia and you're not able to carry oxygen properly, you're going to end up with cyanosis, which is just bluish discoloration of the skin. This picture is not in your lecture, but it's just a fun fact that there is a family which have the genetic deficiency of methemoglobin reductase, and this gives them blue skin. So they're literally called the blue family or blue skin family. Blue people, that's what they're called. They're the blue fugate family. This is just a fun fact to remember that methemoglobinemia is hereditary. That's it for methemoglobinemia. Pretty straightforward. So now this is just a quick table. You've got different enzymes that can have different deficiency results. All right, thank you so much for uploading the slides. I hope whoever needed them can follow along with the slides. Now, so pyruvate kinase is in glycolysis in the glycolysis pathway. If there's a deficiency, you're going to end up with hemolytic anemia. G6PD is part of the second pathway, the pentose phosphate pathway, to regenerate NADPH. If you don't have G6PD, you're going to have oxidative stress, which is going to oxidize your hemoglobin, resulting in Heinz bodies. You'll take this in detail somewhere else in a different lecture. Methemoglobin reductase deficiency will give you methemoglobinemia, where there will be cyanosis or blue discoloration. Yes, hemolytic anemia, the RBCs burst. Hemolysis is breakdown. Hemolysis. Lysis is bursting or breaking down. Hemo is blood. Now, lastly, we talked about the life of an RBC and the membrane and metabolism and function. It gets destroyed. I already mentioned this, so I won't stress on any of these points. It gets removed in the spleen by macrophages or in the red reticular endothelial system, which is an organ system. And they can either die extravascularly in the macrophages or spleen or liver, or intravascularly in the blood vessels themselves. All of these things with globin and protoporphyrin, bilirubin, you'll take them somewhere else. This is just to come to a full circle moment where we started with red blood cell formation and life cycle, and now it has come to red blood cell death, where it will be destroyed and taken out of the body and then recycled. That's it for the erythrocytes lecture. Do you guys want to solve some questions? Just like it's very simple question. What is PEP? Don't worry about it. It was in the table from the lecture, but it's a part of glycolysis that you don't need to know right now. You'll take glycolysis in mole. Okay, so let's answer the first question. Oh, wow. Okay, Okay, so what is the primary role of NADPH? You guys already answered, mashallah. To regenerate, to reduce glutathione, to combat oxidative stress. Perfect. Next question. Do I have a second question? Yes. Okay. In hereditary spherocytosis, the loss of the membrane surface area leads to the formation of spherocytes. Which of the following best explains why these spherocytes are destroyed in the spleen? Yes, B again. Perfect. Great job, guys. They have reduced deformability and become trapped in splenic cords. Okay, we can take a short break before we move on to iron metabolism. What do you guys think? Break? Break, so yes, okay. خلاص, tamam. We'll take a break and we'll be back in, let's say, 5 to 10 minutes. Sounds good? All right, see you guys in a bit. All right, so now I'll be covering the second lecture, which is iron metabolism. Iron metabolism is gonna get a little bit complex, so I just need you guys to focus, and if you have any questions, please feel free to ask. Let's get started. I'll be covering iron itself, iron status and how we measure iron levels in the blood, how the body recycles it, how the body regulates it, and how this can manifest in clinical conditions. Let's begin with the overview of iron. As you guys know, iron is a part of our hemoglobin. It's part of our blood cells. And the main function is to deliver oxygen. It binds oxygen where it carries it to the cells and it releases it. This is the most important component. The second thing is it's also involved in synthesis of ATP as part of enzymes like cytochromes, for example. Now, the excretion of iron, the fact that there is no active excretion of iron in our body, the body does not have a way of getting rid of iron the way it can get rid of other things, except in things like loss of skin and hair, like natural loss of these things, and intestinal epithelium. Other than these processes, there's no active excretion. There's no way for us to actively get rid of iron. This is going to form the basis of the entire lecture. If you understand that we need to be able to regulate iron very carefully in our body. We don't want too little of iron, and we don't want too much of iron because we can't get rid of it properly. Just keep this in the back of your head throughout the entire lecture because it forms the basis of regulation of iron. Now, iron comes in multiple forms. The body has iron in multiple forms. Functional iron, which has a function of carrying oxygen or part of enzymes, enzyme reaction forming ATP or other things. So obviously the functional forms of iron are in hemoglobin, in myoglobin, and enzymes such as these. There's a storage form of iron. There's ferritin and hemosiderin. Hemosiderin is just a broken down form of ferritin, but these two forms are there to store iron in our body. Transport form of iron, which forms the smallest percentage of iron in our body, is in the transferrin. So transfer, obviously transport, this is easy to remember. Transferrin is the transportation form of iron, where it transports it from one place in our body to another. All right, now, yes, I can explain the functions again. Functions of iron are to carry oxygen in the hemoglobin, or as part of enzymes, where it can help in synthesis of ATP, for example, if it's part of cytochrome. It can act as a cofactor. That's it. Main form is the way you know it, which is obviously oxygen transport in your blood. Iron is part of hemoglobin. So I keep saying that, okay, we need to regulate iron very well. Why do we need to regulate iron? Because iron can be a part of this. Sorry, one second. Iron can be a part of this harmful reaction called the Fenton reaction. Iron in the 2+ state can react with hydrogen peroxide, and this can oxidize it to Fe3+. But also, what ends up happening in this reaction is we end up with this right here. If you guys remember high school chemistry, I don't want to give you flashbacks back to that time, but this is a reactive oxygen species. Remember in erythrocyte metabolism where I was saying, we don't want oxidative stress and damage and free oxygen species. These OH groups with this electron right here, these are reactive oxygen species, which are very harmful and damage proteins, lipids, nucleic acids. They bind to them and they damage them. This is why we need to maintain iron at a very specific level in our body because it can be harmful. For example, in this reaction, in the Fenton reaction. You don't need to memorize this reaction. It's never going to be asked about. You just need to remember that iron can be harmful if there's excess amount of it because of things like this, where you can end up with reactive oxygen species as a byproduct of this reaction of iron. So we need to prevent the formation of reactive oxygen species, okay? You don't need to memorize this. Now, where do we get iron from? We get iron from, obviously, the diet, but also the recycling process in our body. The body is constantly recycling it. This makes up the majority of our supply of iron. We have iron, we use it, we recycle it, and we keep using it. Remember, RBCs die. Where does the iron go? It gets recycled into new RBCs. And obviously, we also eat some iron especially in things like meat, but also in vegetables. So dietary iron, the majority of the foods which have high levels of iron are obviously red meats, but also things like legumes and green vegetables, etc. Some foods may be high in iron, they may not be readily absorbed. This is important when it comes to the form of iron and how we absorb iron. One thing to keep note of throughout this entire lecture and for the rest of your medical career is that there's two forms of iron. Fe2+, which is the reduced form, and Fe3+, the oxidized form. So Fe2+, they also call it the ferrous form of iron. So they say ferrous iron. You should know that they're talking about 2+. And how I remember it, just an easy way to remember it is Fe2, ferrous, the two of us, ferrous, and then Fe3+ is ferric. So sometimes in the question, they literally could say ferrous instead of Fe2+, and you're just going to have to know it. The point here is that Fe2+ is the form of iron which we can absorb. It can easily be absorbed in our body. Fe3+, the ferric form, is not as easily absorbed. Okay? Just keep this in mind. We want Fe2+ in the form of ferrous iron. So there are some factors which increase the rate of iron absorption and make it more readily absorbable in our bodies, and there are some factors which will decrease the rate of iron absorption and affect it. Things that can increase iron absorption, like I just said, Fe2+. It will make it more readily absorbable in our body. The second, another thing is heme. Heme is the non-protein, in the form of heme, it's a non-protein form of part of hemoglobin, and it's found in the meat sources of iron. So in meats instead of vegetables. That's where heme iron is found, and that is a more readily absorbable form of iron. Other things that increase absorption are acids, such as vitamin C, solubilizing agents like sugars and amino acids. And these, do not worry about them right now, but we will get into them and you'll understand them by the end of the lecture. But in this table, just ignore them. The things that will reduce iron absorption are obviously Fe3 plus, as we just mentioned, and also inorganic forms of iron, like in vegetables. Alkalis will decrease iron absorption. Precipitating agents like phytates and phosphates and tea will decrease iron absorption. And then these also, don't worry about them because we will talk about them in this lecture. Now, all right, okay. Okay, there we go. So the next segment is assessing iron status in our blood. So how do we know if somebody has enough iron? How do we know? We take a blood test and we measure certain parameters. You don't just go in and you look at the iron and khalas. It's a bit more complicated than that. We have four things that we look at. Serum iron, obviously, which is just iron in the blood. Ferritin, which remember from previous slide, what is ferritin? It's the storage form of iron. Transferrin slash TIBC, total iron binding capacity. This just shows you how much transferrin there is. Remember, transferrin is a transport form of iron. So how much transferrin is there available to carry around iron in our body? And lastly, transferrin saturation. And I'll explain the difference between transferrin or TIBC versus transferrin saturation. Let's begin with serum iron. Serum iron is just straightforward iron in your body, iron in your blood. Ferritin is the intracellular iron storage form of iron. So ferritin, we measure it to know how much iron is being stored. If ferritin is low, obviously this means that this person does not have enough total body iron stores. Then we have transferrin and total iron binding capacity. So transferrin or TIBC is to show you how many transferrin molecules are available to bind and carry iron in our body. TIBC is just an indirect measurement of transferrin, but this is there to show you not how much iron is in the body or iron is being stored. Rather, it's showing you how much transferrin molecules. Transferrin. It just means that there's transferrin available to bind. Did connection cut off? Can everyone still hear me well? Okay, I'm gonna assume that nothing happened. All right. So, okay. Transferrin saturation, the fourth form, is the amount of, what's the difference between 3 and 4? I'm getting to that. Transferrin saturation, we just talked about transferrin and TIBC. Transferrin saturation is the amount of iron that is bound to the transferrin. Okay? So let's think of it like this. Transferrin is the bus, okay? Transferrin saturation is how much iron is in the bus, how much iron is being carried. Transferrin is just the empty bus. How much availability is there for iron to be carried? So it's like the seats on a bus is transferrin, and transferrin saturation is the iron people that are sitting in the bus. Transferrin is just the molecules that are available to bind and carry iron. Saturation is how saturated it is and how much iron is bound. And you calculate it by iron divided by the total iron binding capacity. So think of it, okay, how much transferrin is there, or TIBC? What is the capacity of our body to carry iron? Saturation is how much iron there really is. I'll explain this as a form of iron deficiency anemia, and it's more likely to make sense then. Can you explain TIBC? TIBC is total iron binding capacity. It's the capacity for the body to carry iron. How does it carry iron? By transferrin. So how much is there? How many molecules of transferrin are there to bind and carry iron? To potentially bind and carry iron. It doesn't mean how much iron is there. It means the capacity for the body to carry iron via measuring how much transferrin is in our blood. Saturation is how saturated the transferrin is, how much there is iron on the transferrin, how much is being carried itself. They're two separate things. Okay, perfect. So now think of it like this. If there's iron deficiency, how is it going to affect these parameters when you take the blood test from somebody? Let's say you're the doctor, you're looking at somebody's blood test. If they have iron deficiency, what is going to happen to their iron, their ferritin, and their transferrin? And okay, let's start with serum iron. Any guesses for what's going to happen to the serum iron? Obviously, it's going to decrease. Obviously, there will be less serum iron. This is straightforward. What about the ferritin? it's also going to decrease because you don't have stores of iron. So ferritin is also going to decrease. بالظبط. Now, what about transferrin? Increase. MashaAllah, great job, guys. It will increase. Why? Because the body is going to go and try to look for more iron to carry and transport in our body. It's going to start making more transferrin, and the transferrin itself is empty. It's not bound to anything. So transferrin itself is going to increase because it's not binding iron. It's just looking for the iron to bind. This is what's going to happen in iron deficiency. So keep in mind, because you would think, oh, everything would decrease. No, because transferrin, we're not measuring iron. We're measuring how much transferrin there is to bind. So, khalas, you guys got it. Shall I understood the concept? Let's move on. Now, next segment is about iron recycling. We said we can't get rid of it, so we recycle it. How do we recycle it? Now, iron, we consume it in our food. It's in our intestines. We absorb it through the enterocyte or the intestinal cell. And then it moves, gets carried by transferrin. TF is transferrin, gets carried by transferrin in the blood, where it goes to the bone marrow in a developing red blood cell. It gives the developing red blood cell the iron. Now the red blood cell has matured and developed because now it has iron. It circulates in the body. After 120 days, it gets destroyed by a macrophage in the spleen, where after it gets destroyed in the macrophage of the spleen, it gets broken down. It gets broken down. The hemoglobin gets broken down. The heme, the globin, the iron, everything gets broken down, and we need to recycle. Iron leaves the macrophage, gets carried around by the transferrin in the blood, where it goes to the liver. In the liver hepatocytes, the iron again gets recycled, leaves into the blood, where it goes back to the developing red blood cells in the bone marrow and gets used again. This is to show you that we need to constantly recycle iron. We don't just use it and then lose it. We have to keep recycling it. So this means that most of the iron in our body comes from the recycling of old red blood cells, okay? After they're broken down in macrophages. It's when we get too much iron that we don't want to keep recycling, we can store it in the form of ferritin. Now, how does iron leave the macrophage and enter the blood? Through a very important channel called a ferroportin. Port, like a port where something moves through. So think of it like ferroportin is the port that iron moves through. It's the channel in the membrane of macrophages where iron can leave. This is a really important channel because it's going to become very important when we talk more throughout this lecture. So ferroportin is in macrophages, but it's also in liver cells and it's in gut cells. That's how iron is moving out of these cells, out of the enterocyte, the macrophage, and the hepatocyte. Just keep ferroportin in your head. So, in addition to what we just mentioned, there are some plasma proteins which help to store iron or carry iron. We have two of them. We have haptoglobin and hemopexin. Haptoglobin will grab the free hemoglobin after RBCs are broken down. Haptoglobin will bind to the hemoglobin and will form the haptoglobin hemoglobin complex, which is then taken in by macrophages, and then macrophages do what they will with them, whether it's to store them or get rid of them. On the other hand, hemopexin will grab the free heme. Heme is the non-protein part of hemoglobin, which carries iron. It will form the hemopexin heme complex, and then this will be taken up by liver cells or hepatocytes. So this is just to show you that macrophages will take up the haptoglobin with the hemoglobin. Liver cells will take up hemopexin and heme. Why do we need to get iron from our food if we already have iron before? We already have iron before, but there's a small amount that we still need to add to our bodies. We excreted. I mentioned that we excreted through the loss of hair and skin and intestinal epithelia. There are some minute form parts of iron that are lost every day that we need to supplement, obviously, with the food we eat, and it's found in the food we eat, so we just use it. But the majority of our iron is recycled, so we don't need to constantly take in iron from outside. If we have normal iron recycling pathways and processes, we can function normally and just get some supplementation of it in the meat that we eat and the vegetables we eat. We lose iron through loss of skin, loss of hair, loss of blood, loss of in the intestinal epithelium. There are ways that you can lose iron. The reason why I'm stressing on the fact that you don't have active excretion is that when there's too much iron, this is a problem because you can't get rid of. Can you explain both proteins? Yes. There's haptoglobin and hemopexin. Haptoglobin binds hemoglobin. Just from the name, haptoglobin, hemoglobin, you can put them together. Haptoglobin binds hemoglobin, and this forms a complex which is taken up by macrophages because they have receptors for haptoglobin hemoglobin, the macrophages. Hemopexin, on the other hand, heme binds heme. Okay? Hemopexin binds heme. This forms hemopexin heme complex. This complex is recognized by liver cells and taken up by the liver. Why is this important? This is just to show you that these proteins prevent iron loss in urine by keeping hemoglobin and heme bound in the blood, so it maintains levels of iron in our bodies. We don't want to lose these. We don't want to lose hemoglobin and iron, so we have ways to keep them. If hemopexin takes the heme, does heptoglobin only take the globin? So haptoglobin takes the whole hemoglobin. Don't overthink it. Just don't overthink it. Take it at face value. Haptoglobin takes the hemoglobin. Hemopexin takes the heme. Okay? I hope this cleared it up. Yes, perfect. All right. So now we've arrived to the most important part of this lecture, regulation of iron. And I need you to listen very carefully and focus. If you have an issue, just mention it in the chat. Now we'll introduce a new player, hepcidin. Hepcidin is a hormone that is made by the liver. It is there to control how much iron is in the blood, is released into the blood, and is absorbed by the body. How does it control this? Because it acts like a lock on the channel that we mentioned before, the channel which allows iron to leave cells, ferroportin. Hepcidin is a lock on that channel. It will block that channel when it needs to, and it will open that channel when it needs to. Okay? So what is controlling iron absorption and movement of iron out of the cells? Hepcidin. By controlling ferroportin. So let's start with how hepcidin does its job. Hepcidin, when hepcidin levels are high, it will go and block ferroportin. It puts the lock on ferroportin. This stops iron from leaving cells. Tells the body that خلاص, we have enough iron. We don't need more iron to be absorbed. Stop absorbing iron and stop releasing iron from the stores that we have. This keeps iron inside the cells, and it also keeps iron away from being absorbed in the intestines. Okay? Because sometimes خلاص, we have enough, and I told you we shouldn't have more iron than we need. So it tells the body to stop absorbing it from the food that we eat. However, when hepcidin levels are low, there's not going to be a lock on the ferroportin. Ferroportin is going to stay open. Ferroportin is going to stay open means iron can keep entering the blood. It can be absorbed from our intestines, and it can bring us back to normal levels if we don't have enough iron. So hepcidin is high when we have enough iron. We don't need more. We need to keep it in the stores, or we need to stop absorbing it. That's when hepcidin levels are high. So it can stop this process of increasing iron. When we don't have enough iron, hepcidin levels are low because we don't need anything to block ferroportin. We need ferroportin to stay open. Ferroportin stays open, and therefore more iron enters the blood. More iron is being transported and moving around to make RBCs, etc. Is this clear? The function of hepcidin? Okay. Yes, okay, perfect. Now, let's move on to this is again just to look at what hepcidin does. See, remember this diagram? Hepatocytes, the liver cells can produce hepcidin to go block this iron from leaving macrophages by blocking ferroportin, and also by blocking the ferroportin in intestinal cells to stop absorbing it. So this is where hepcidin comes in this life cycle. Now, let's take a look at the intestinal cell. Remember here how this process goes from intestines into the intestinal cells into the plasma. Let's go and zoom in into what exactly is happening in this process. Can you explain the diagram again? This diagram, the previous one? This one? Yes, okay. Here, iron is in the intestines because we consumed food, there's iron in the food. This iron gets absorbed into the intestinal cell, enterocyte, where it will be released from the enterocyte into the blood, where it binds to transferrin. Transferrin takes this iron and takes it to the bone marrow, where it releases it. Iron goes into the bone marrow, into the developing red blood cell, making it a mature red blood cell that's ready to go and function on its own. Circulating red blood cell circulates for 120 days, where it's then taken up after 120 days and its life cycle is over. It goes to the spleen, the macrophages destroy it. Now we still have iron. The iron, after we break down the the aged RBC, the iron is then going to be released again by ferroportin into the plasma, where the transferrin is going to take it to the liver, and the liver is going to release it to the blood, and the blood is going to go and put it back in a developing red blood cell. This is the recycling process of iron. This is the external when we consume iron from food. And hepcidin is released from the liver cells or hepatocytes. Hepcidin goes and blocks any exit of iron from macrophages and blocks the exit of iron from intestinal cells. So it stops iron from going into the blood, basically, when we have enough iron. Doesn't all this happen in the duodenum? Which part? This is the intestine, this is the intestinal cells, this is the duodenum. This is where iron absorbed, yes. But the rest of this is happening in the spleen, the blood, the liver. If you're talking about this, then yes, I'm talking about the intestines and the duodenum here. We can get started with this diagram now. You're welcome. All right, great. So this diagram, I know it looks very, very overwhelming, but we're going to break it down step by step. Before we start before we start explaining the diagram, just keep in mind there's two sides of the intestinal cell. This is a cell inside the intestine, okay? There's the luminal side, which is the lumen of the intestines, and there's a basolateral side, which is facing the blood. So, okay, I'm going to start with the luminal, and then I'm going to talk about the basolateral side. So the luminal mechanisms, which is facing the outside, this is where the iron we consume from food, that's where it is, and that's how it gets absorbed. So in the form of heme, when we eat it in the form of heme, which is mostly in like things like red meat, heme is absorbed into the enterocyte or intestinal cell via a heme transporter, which then is this enzyme HO1, which is heme oxygenase 1, is going to take out the iron from the heme. Okay, this is one way. The second way is the iron 3+, which we have in food. If it's in the form of iron 3+, there's a channel enzyme called D-cytochrome B, or D-CYP, which is going to convert Fe3+ to Fe2+. If you remember, I said the way we absorb iron is Fe2+, not Fe3+. So we need something to convert it. So now we've converted Fe3+ to Fe2+. That's when Fe2+ can be absorbed into the cell via DMT1, which is divalent metal transporter 1. So DMT1, Fe2+ goes through a DMT1 into the cell. Now we've got the iron absorbed in two ways, from heme through a heme transporter and from DMT1. Is it safe to store iron as Fe2+? Usually we convert it back to Fe3+ to store it, but for absorption, we need it we need it to be Fe2+. For storage, it's usually transferred back to Fe3+ for storage form. Now, in the cells, we have ferritin, as we said, the storage form. Ferritin has it can act on the Fe2+ and convert to Fe3+, so it can be stored. Where it can be converted again to Fe3+ in ferritin, this part that I was getting to. So now we absorbed it in Fe2+, converted to Fe3+ to store it in the form of ferritin. Ferritin has enzyme activity. Now we've reached this point. Is this part clear so far? Because I know this can look confusing, but it's really not that complicated if you look at it one by one. Now, okay, perfect. Now we've got the stored iron. We want to release it into the blood. This is we haven't released it into the blood yet. So now we're going to move on to basolateral transport. We're done with the luminal side. We're in the basolateral side. Now the iron, we want to release it into the blood. How do we release it to the blood? We already said this. Ferroportin. Ferroportin is a channel that allows iron to leave intestinal cells. Ferroportin allows the exit. Fe2+ is now in our blood. Now what if we have enough iron? Hepcidin is going to come and block ferroportin. Okay. Hepcidin is the one that comes and blocks ferroportin if we have enough. If we don't have enough, ferroportin stays open. Now, there's another protein on the outside of the membrane of the enterocyte called hepastin. Hepastin will convert Fe2+ to Fe3+ once again to bind to transferrin, so that transferrin can go and carry Fe3+ and take it around the body. So the point of Fe2+, Fe3+, don't get confused. The only thing you need to remember is that Fe2+ is the form that we need to absorb it. We can store it as Fe3+, we can transport it as Fe3+, but to absorb it, we need Fe2+. This is why there's this enzyme D-cytochrome B, D-CYP. Everything else can go smoothly other than that. All right, this is just a table highlighting everything I just said. So the function of every single one of these little channels and enzymes and ions. We have D-cytochrome B, DMT1, heme oxygenase. This is just for you to come back to and remember. But I already explained the entire thing. This is just for your own, to make it easier for you. So it leaves as Fe3 plus again. Yes, it leaves as Fe3 plus. Because what converts it? Hepastin. Hepastin changes Fe2 plus to Fe3 plus as it exits the cell, preparing it to transport in the blood. Got it. Appreciate it. You're welcome. All right. So this is just a table for your own reference. If you forget something, just come back to this table. And hepcidin. Hepcidin doesn't convert to Fe3 plus. Hepcidin blocks ferroportin. Hepcidin is just to block ferroportin and stop absorption. Yeah. Great. Alhamdulillah. Hepcidin is just there to block this. You're welcome. All right. Is there another question? Difference between apotransferin. Okay. So apotransferin, I'm glad you brought that up. I was about to forget to mention that. So apotransferin is just the empty transferrin. Apo, empty. Empty transferrin. Once iron binds to transferrin, it becomes hollow transferrin. Okay? Apotransferrin is empty transferrin. Hollow transferrin is fully loaded transferrin with iron. So if you remember, just to link the ideas together, when we talk about transferrin total iron binding capacity, or TIBC, in the blood tests, we're looking at apotransferrin, which is empty. How much there is to carry. How come we need hepastin when it's stored as Fe3 plus? That is a really good question. 1 second, let me just get rid of the chat box. So to, again, leave the cell, it needs to be in the form of Fe2 plus. So to absorb it, it needs to be in the form of Fe2 plus. To leave, it needs to be in the form of Fe2 plus. Afterwards, we need to convert it back to Fe3 plus to be carried. So see how much conversion there is. This is where I don't want you guys to get confused. It gets converted in order to be absorbed. Fe2 plus is the absorbable form. Then when it's inside, it can be stored as Fe3 plus. It can be converted back to Fe3 plus. But then once it wants to leave again, it needs to be converted back to Fe2 plus. So the absorbable and the form of iron which enters and exits is in the form of Fe2 plus. Got it. Thank you. Great. All right. So if this is clear, okay, 1 second. So ferritin converts 2 ways. Yes, it does convert 2 ways. So when transport, it will be Fe2 plus. When it leaves the cell, it will be in the form of Fe2 plus. When it binds to transferrin, it will be Fe3 plus. Since we converted it by hepastin. Okay. This is just the written form of everything I said, so I'm not going to go through this again. This is just the written form if you need notes. But I already explained this. All right. If it's clear, I'll move on. Okay. Tamam. Perfect. Now, yes. Okay. Now, let's go back. Just to reiterate, because this idea it can get confusing, and I just want to make sure it's repeated enough to stick in your heads. Hepcidin is there to control levels of iron. When there's too much iron in our body, hepcidin is released. When hepcidin is released, it binds to ferroportin and blocks it. What tells our body that iron stores are full, or there's enough iron in our body? Holotransferrin, which is full of iron, is going to go to the liver cells and tell the liver cells, no, we have enough iron. When the liver cells see that there's enough iron because of the holotransferrin, they're going to start producing hepcidin. Hepcidin goes and blocks ferroportin to prevent release of iron. On the flip side, when iron is low, hepcidin will decrease because we don't want anything to block its absorption. So khalas, liver stops producing hepcidin because we need more iron. Ferroportin is now more active since something is blocking it. And iron enters the blood from the intestines, from the macrophages, and from the liver cells, where we can increase our iron levels once again. This is the function of hepcidin and how it functions. So this is reiteration. Hepcidin goes up when we have enough iron. Khalas, hepcidin is there to tell the body, enough, we have enough, because transferrin is full. In other cases where hepcidin increases, it also increases in inflammation and infection. This we'll get to in a minute, why this is important. On the flip side, hepcidin goes down when you don't have enough iron. We need more iron, so hepcidin is going to go down. In what cases do we need hepcidin to go down so we can absorb more iron? When there's anemia, when there's low oxygen, when we need to absorb more iron, when there's high erythropoietin. Why is this important? Is that if hepcidin levels are too low for too long, the hepcidin absorbs too much iron. Okay, so there needs to be a balance of hepcidin and our iron levels. We don't want hepcidin to be too low for too long because then the ferroportin is open and active. We absorb way too much iron. This will lead to iron overload and a disease that we'll talk about in a few minutes. When body iron is low, what stimulates hepatocytes to limit production of hepcidin? That is a really good question. Let's go back to the slide. When hepcidin, here. When body iron is low, what tells hepatocytes? The fact that transferrin is now empty. See how hepcidin was triggered to be released by the cells because holotransferrin was full. Holo means full transferrin or fully loaded transferrin. When there's holotransferrin, hepatocytes can sense the holotransferrin and it goes like, oh, there's so much so much iron in our body since transferrin is full, so let's produce hepcidin. The opposite happens when iron is low. When iron is low, there's no holotransferrin because the transferrin is not fully loaded. There's not enough iron to fully load transferrin. If there is no holotransferrin for the hepatocytes to see and detect, the liver cells detect that there isn't enough iron because there's no holotransferrin around. There's no fully loaded transferrin. So it's going to stop producing hepcidin. What is the function of erythropoietin? We're going to get that in a minute. Erythropoietin is there to tell the body to produce more red blood cells. It's the hormone which makes the body produce more red blood cells. And it's mentioned here because there's high erythropoietin, which means we need to make more blood cells. If you need to make more blood cells, you need iron. So why would you need hepcidin? You need hepcidin to go down so that you can have more iron. Erythropoietin is definitely going to be explained to you in your hematology lectures, I think, especially. But if not, then it's just there to it's the hormone which tells the body to make more blood cells. Okay, if everything is clear. Any more questions? Okay. Awesome. Now, another part of this regulation of iron. I know this is a long lecture, it gets complicated, but just you guys are almost done. Erythroferron. Erythroferron is a signal that is sent out by RBC producing cells or like immature RBCs. This tells the liver to reduce hepcidin so more iron can enter the blood, and this especially happens during anemia. So in anemia, when there's not enough RBCs, the kidneys are going to release erythropoietin, which you just asked about, erythropoietin. Then erythropoietin tells the erythroblasts or immature RBCs to make more red blood cells and more erythroferron. So more erythroferron, less hepcidin. So if you don't have enough red blood cells, if you have anemia, your kidneys will release erythropoietin, EPO. EPO is going to go and cause an increase in release of erythroferron. Erythroferron is going to tell the liver to stop making hepcidin so that we can have more iron. Okay? Just understand the sequence of things. EPO or erythropoietin is there to increase red blood cells. If we want to increase red blood cells, we have to increase erythroferron so that it can tell the liver to stop making hepcidin, so that iron can increase. This diagram was in your lecture. I don't think it's just everything we've been talking about. It's not that important. This is just to show you what is affecting hepcidin synthesis. So what is blocking hepcidin synthesis? Erythroblasts and erythroferron, not enough oxygen or hypoxia, erythropoietin, and forget about the last one, metryptase. Can you repeat erythroferron? Yes. Let's go back. Erythroferron is released when the body needs to produce more red blood cells. What triggers its release? Erythropoietin. They're both just hormones or molecules that are released to make more blood cells, to signal to the body to make more blood cells. If you don't have enough blood cells, your kidneys are going to make erythropoietin, EPO. Erythropoietin is going to trigger the release of erythroferron. Okay? Erythroferron is just a signal molecule. Erythroferron is going to tell the liver to stop making hepcidin. Because what does hepcidin do? It blocks iron absorption. Now we need iron absorption. So Erythroferrin tells the liver to stop making hepcidin. When we have less hepcidin, we get more iron absorption. More iron absorption, better production of red blood cells, which we need. This is what erythroferrin does. So think about it like this, like you're probably going to be asked about it in the form of what increases in what situation and what decreases in what situation. So when erythropoietin goes up, EPO, then erythroferrin goes up, ERFE, which will cause hepcidin to go down, which will cause iron to go up. That's just the sequence that I want you to focus on. Okay? All right. We're almost to the finish line. Now, we talked about how the body absorbs iron, but let's go into a little bit more detail. So as we said, we don't want too much iron. So we need to carefully control how much iron is in our body. We said one way is through hepcidin and ferroportin blocking, but we also have other mechanisms. Now, cells have transferrin receptors, TFRs, transferrin receptors, that will bind to transferrin. This is the absorption of iron, okay? Once transferrin binds to the transferrin receptor, actually, let me explain on the diagram. Look here. This is a transferrin receptor. This orange thing with two legs, this is a transferrin receptor. This green olive looking thing is transferrin, and the purple is the iron. So this is transferrin, hollow transferrin. Remember? Hollow transferrin. Hollow transferrin is going to bind to the transferrin receptor. Once it binds to the transferrin receptor, it will be taken into the cell by making an endosome. So the cell membrane is going to pinch out and make a little endosome inside. This is the endosome with transferrin receptor bound to transferrin. Now, the endosome is going to get acidified by, there's going to be a proton pump that pumps hydrogen ions inside. What's the purpose of this? The hydrogen ions are going to cause the release of the iron from the transferrin. See how the purple dots, which is iron, have been released from the transferrin? Now, the iron is detached from the transferrin. Iron now leaves the cell through DMT1. Remember, divalent metal transporter 1. It releases the iron. Iron is now in our cytoplasm, where we can use it or store it or transfer it or do whatever we want with this. This is how iron goes from being bound to transferrin to being eaten up by the cell so that it can be used. And this is then recycled, where transferrin receptor goes back to the surface of the membrane. The process can happen again and again. And in this way, in this process, you can prevent too much iron absorption by controlling the amount of transferrin receptor. So when the body has too much iron, it will remove the transferrin receptor from the membrane so that we can stop absorbing iron. This is just how the iron is absorbed into the cells. Now, next, we talked about transferrin and absorption. Let's talk about storage. Cells store iron in the form of ferritin. Let me answer a question. I got confused. Can you please repeat it? Yes, I can repeat it. 1 second. I'm confused. This is another way of iron metabolism. This is not another way. This is just how iron is, like when we go into more details about the actual absorption of iron into the cells. So how does iron enter cells after it has been bound by transferrin? Remember after we absorbed iron in the diagram that, okay, there. Remember how now we absorbed it through the intestines and now it's in our blood through transferrin. So what do you do with the transferrin once you want the iron to go into the cells? Now you want to use the iron. The transferrin then binds to transferrin receptor, where it's engulfed into the cell, into an endosome, where then the iron is released from the transferrin. The iron is taken into the cells so it can be used, and then the transferrin goes back as apotransferrin. This is just the recycling process and how it does that. Don't worry about the tiny details. Just in the got it. Perfect. What about what did you get it? Do you want me to explain it again? All right. Okay, great. Now back to ferritin, the storage form. Cells store iron in the form of ferritin, because so you can keep iron stores, obviously. And where is it mostly stored? In macrophages and liver cells or hepatocytes. Again, Fe3 plus is the way that we store iron usually, because ferritin converts Fe2 plus to Fe3 plus to store it, and it can hold a lot of iron ions, one molecule of ferritin. Now, once you want iron again, you will break down the ferritin. So obviously, because you want to break down the stores of iron. You break down the ferritin, And in the process of breaking down ferritin, it becomes hemosiderin. This is partly broken down ferritin, the leftover form. So this is just to give you to understand the names. Ferritin versus hemosiderin. They're both storage forms of iron. And this is, again, regulation. So how do we regulate iron storage and how much iron we absorb? By transferrin receptor. If we make less transferrin receptor, you stop absorbing iron. If you make more, you start absorbing more. Very straightforward. Transferrin receptor is the thing that's absorbing it to begin with. Okay? Are we ready to move on to the diseases? What's the difference between the first cycle and the second cycle? Like, what's the difference in their functions? Cycle? What do you mean by cycle? I'm not sure. What's TRF1? Transferrin receptor. Transferrin receptor 1 is just the receptor that binds to transferrin so that it can be absorbed into the cell. Iron cycle and cellular absorption. So I think you mean this part. This part is the absorption from the intestines. This is the enterocytes specifically. This is what happens. This is how we absorb iron from food into the blood. This second picture, the diagram that I explained with the transferrin receptor, this part is how now we've absorbed iron from our food and it's bound to transferrin. How do we get it into cells? So this is the cells of the liver, the cells of like macrophages, the bone marrow. This is how we use the iron that we've now absorbed, how it enters the cells. Okay, you're welcome. All right, this is the last part. We're almost done, guys. Just stick with me for a bit more. Now, everything I've been talking about throughout iron metabolism is for the purpose of recycling iron and regulating the amount so we don't end up with too much iron. Why? Because we cannot excrete when we have too much iron. It's a problem. We cannot just get rid of it as simple as you'd think it is. We're trying to avoid iron overload. So when there's excess iron storage, this happens when the body's rate of iron intake is less than loss. The body stores excess iron as form of ferritin and hemosiderin, as we know. Now, if there is overload where there's too much, what can happen? First of all, what is it caused by? What can cause this states of iron overload? Normally, we are not supposed to have iron overload. Your body knows how to regulate iron. It can happen in certain disorders, genetic disorders, like number 1, hereditary hemochromatosis. Hereditary means genetic. Hemochromatosis means, let's just take it as too much iron in your body. Or it can happen secondary to chronic anemias, when anemias like thalassemia, beta thalassemia go on for, like, they're chronic, they become chronic, long-term anemias. This results in hemochromatosis or iron overload. How? Number 1, first of all, you have too much. When you have excess iron, this triggers the Fenton reaction. Remember the chemical reaction which produced a reactive oxygen species? These reactive oxygen species will oxidize lipids, proteins, nucleic acids, and heme, which will lead to cell damage and death. Cell damage and cell death in the liver, the heart, the pancreas. This is bad. You don't want free oxygen species. In terms of hemolytic anemias, the bone marrow is working overtime because there's hemolytic anemia. Your blood cells are broken down. Your bone marrow is trying to make more and more and more to make up for it. So this ends up making more immature red blood cells, which are erythroblasts. And remember what we said about erythroblasts, they release erythroferron. What happens when you release erythroferron? You inhibit hepcidin. If you inhibit hepcidin, which is the iron blocker, it keeps ferroportin open. So iron keeps coming in and in and in. You end up absorbing way more iron than can be recycled and stored. You end up with iron overload, which is iron that you can't really use because there's a root problem, which is the hemolytic anemia. That's what's causing the bone marrow to work overtime and make more erythroferron, less hepcidin, more iron. So you end up with iron overload. So blood donation would help in iron overload cases? No, it wouldn't, because you have too much iron. And if you get a transfusion of red blood cells, you would increase more iron. If you're talking about the hereditary hemochromatosis where you want normal blood cells, the treatment of it, I don't believe is a blood donation or a transfusion. Like, it's specific management. It's a genetic disorder in any case. Okay. So if that's clear, Okay. All right. So the point is, if it's a genetic cause or it's from chronic anemia, iron overload is bad and it can harm your organs because of the reactive oxygen species. You don't want this. And this happens because the body keeps absorbing iron when it doesn't need it. All right. Now, we're done with iron overload. The second thing that I want to discuss is anemia of chronic inflammation. This is the last part. You guys are almost done. Let's just finish it up. Anemia of chronic inflammation. Let's break this down. Chronic means something that's been going on for a long time. Inflammation is any disorder which is causing inflammation in your body for a long time, such as chronic infections, such as malignancies, cancers, and autoimmune disorders and dysregulation. All of this will result in constant inflammation in your body, an inflammatory state. Okay? What this does is it's going to activate your immune cells, your T cells, and your macrophages. Once these are activated, they will release certain cytokines or chemicals such as TNF alpha, interferon gamma, and interleukin 1. Don't memorize these, don't worry about them right now. The point is your immune cells are being triggered by inflammation and they're releasing these cytokines. And what these cytokines do is they will go to the bone marrow and they're going to suppress erythropoiesis, which is the production of blood cells. It will tell the bone marrow to stop making blood cells. It will also go to the kidney and tell the kidney to stop making erythropoietin or EPO. Obviously, what is the outcome of this? You're going to have less red blood cells, which is what anemia is. So this is how diseases of chronic inflammation, such as infections and cancer and autoimmune diseases, will lead to your bone marrow making less blood cells by the inhibition of erythropoiesis, and also inhibition of erythropoietin from your kidney, which the end product is less red blood cells made. This is how you end up with anemia. Another way you end up with anemia is that all of these triggers will cause your immune cells to release interleukin 6. Interleukin 6, this is the important one that you need to remember. Interleukin 6 goes to the liver. It's a chemical, it's just a chemical. IL-6 goes to the liver and tells the liver to do what? To raise hepcidin. Now remember what hepcidin does? It blocks ferroportin. So if ferroportin is blocked by hepcidin, that means you're not absorbing iron. It's going to affect the intestinal cells and tells them to stop absorbing iron. And it's also going to go and block ferroportin in the macrophages and liver cells and to keep iron stored instead of releasing it into the blood. So this is going to result in less iron absorption from your intestines and less iron released from your stores. All of these three things, the red blood cell production being inhibited, and also the iron absorption being inhibited and iron release being inhibited, will result in anemia, which is a decrease in hemoglobin and red blood cells. This is what happens in anemia of chronic inflammation. Okay, I tried to break this down to make it as simple and straightforward to you. Chronic inflammation will trigger your immune cells to go and suppress erythropoiesis in the bone marrow, suppress EPO from the kidney. These two things will result in less red blood cells production. At the same time, chronic inflammation will lead to an increase in IL-6, which will tell the liver to make more hepcidin. Hepcidin blocks iron from being absorbed and released. This all comes together to give you anemia of chronic inflammation. Okay? This is just in written form, everything I explained, since I explained on the diagram. This is the written form of what I just said. Is that clear? We're done. We just have questions left. When will video be uploaded to YouTube? Inshallah, as soon as possible. I think if not today, then tomorrow by the G&D team. No, not really. Last point. Okay, let's just go over this again. Which point? The hepcidin point? Okay, I'll just go over this one more time. Like the whole thing. Okay, no problem. I'll explain it again. So there is a type of anemia called anemia of chronic inflammation. Why it happens is very relevant to everything that we just talked about. So if you understood the lecture, this part is going to come easy to you. Why does anemia of chronic inflammation happen? It happens because there is a situation of chronic inflammation. How does chronic inflammation happen in our body? By infections, by malignancies, cancers, and autoimmune diseases. You have inflammation in your body from some cause, whatever it is. This inflammation activates your immune cells, such as T cells and macrophages, to release chemicals called cytokines, okay? These chemicals are all of these here. TNF alpha, and don't worry about them, the names of them. This is just to show you that the immune cells are releasing them. And these cytokines will have three effects. Number one, they will suppress production of blood cells in the bone marrow. They just suppress it. They stop production of blood cells. Second thing they do is they suppress erythropoietin released from the kidney, which also stops blood cell production, leading to the outcome of less red blood cell production, leading to anemia. This is one way. These are the two ways. Third way is interleukin 6 production, which goes to the liver and tells the liver to make more hepcidin. Hepcidin goes and stops iron absorption, stops iron release, leading to anemia again. So in these three ways, the hepcidin, the erythropoiesis, and erythropoietin inhibition, you end up with anemia of chronic inflammation. Like the whole thing, anemia. Is it clear now? Anemia is just the decreased hemoglobin and red blood cells, and this is why it happens in case of chronic. Oh, I get it. Either you get anemia from low blood or iron. Yes, yes, exactly. This all comes together to give you the anemia because of the chronic inflammation. You're welcome. All right, now let's just answer one question. I think I just put one question at the end of this, and then you guys are off to go and enjoy the rest of your day. Okay. Now, a 45-year-old man with chronic anemia is found to have low serum iron, low transferrin saturation, but high ferritin. Which regulatory hormone, which is secreted by the liver, is most responsible for these findings? Very easy question. Yes. You guys are all right. It is hepcidin B. Hepcidin is responsible for blocking iron absorption in the case of this chronic anemia. Okay, I think that's it. Yes, that was it. I hope you guys understood. I really hope this clarified these lectures for you. Please scan the feedback form where you will find a small quiz, again, if you want to test yourself on the lecture. It will be uploaded to YouTube. If you need any clarifications, please feel free to reach out. My email is this on the screen. Please feel free to reach out at any time. And I'm fully available to answering your questions. I just really hope this was a beneficial PAL session. You're all very welcome. If you have questions right now, please feel free to ask them as well. But that's it for me. Thank you guys.